Hi, Dr. Lee. Can you explain how cancer actually develops inside of people's body?
Okay, so the main reason that cancer is developed is when the cells is uncontrollably reproducing and there's way too many, or this process is also known as mitosis. Also, this is kind of a little bit off topic, but some gene therapies that are used for treatment, they actually use something called viral vectors. And basically, what that means is they're inserting these viruses, these retroviruses, to try to find areas where they can get rid of the cancer. But unfortunately, sometimes it can go wrong. So some gene therapies has been suspended from trials because they accidentally gave patients cancer. But how it works is when you have the virus and you do a viral vector and you insert it, basically what your goal is you're trying to get the virus to insert their own DNA sequence into your chromosome. But sometimes if it lands near a gene that is responsible for cell division, it could potentially cause cancer because if it lands there, it messes up the way the gene is functioning, and it could cause uncontrollable cell division. But usually this is pretty rare because most of our chromosomes and DNA is mostly blank junk DNA, which has no effect. There's only a few spots where there's actual genes lined up there, and there's also regulatory regions. So if the virus lands on the gene or a regulatory region is basically part of the DNA that helps the gene function. So if it lands on either of those two things, it could potentially cause cancer. There's also another type of gene that helps your cells, your just regular cells, function. If the virus lands on that, it's probably fine because it's just one cell, so that cell will probably die, but the rest of your body can still function since it's just one cell. But yeah, usually when they're doing the viral vectors, the viruses are pretty random, so you don't know where they will land. So this is why it's pretty risky treatment, and they're still developing how this treatment will fully work.
I guess that's why you have a lot of clinical trial for the gene therapy.
Yeah.
But now there's all the research studies. So explain to me a little bit. How do you actually insert a gene into a DNA?
Okay, so in the process of gene insertion, there's 3 ways to do it, but there's only really 2 ways for humans. The first way is kind of just to insert a gene into bacteria, because bacteria are prokaryotic cells. So this basically what that means is the cell does not have a nucleus, which our cells do. So this could only work for bacteria because they're prokaryotic. And basically what you can literally just insert it by spreading it or trying to put it into the this saline, and then you can insert it to the bacteria because it's a single cell organism. It's not very complicated at all. But for a human, if it's eukaryotic, which means it does have a nucleus, the way to insert a gene, first of all, the first way is if the DNA cell itself is hydrophobic, which means means it does not attract to water. So when you insert a gene, it has to pass through the cell membrane. And the cell membrane, basically, it has these hydrolipids. They're like these things with heads and tails, kind of like a sperm, but the tails are hydrophobic and the heads are hydrophilic. And what that means,
Wow.
hydrophobic does not like water, the heads like water. And so there's 2 sides of it. One side is like this, the other side is like this. Like this. So the tails are always facing each other, which means the middle part is hydrophobic. And so if this DNA cell is hydrophobic, it can pass through easily. If it's not, it cannot. But if it's not hydrophobic, it can pass through the protein channel to also get through and insert itself. So that's the second way. And yeah, that's how you insert a gene.
Well, thank you so much, Dr. Lee, and give us a very good education. Well, I'm looking forward to your more channel. Thank you, Dr. Lee.
Thank you.